Hello and good afternoon. Um, Today we're going to explore IPERION HS and archaeology and what it can do for you as archaeologists working in the field. I'm going to start by sharing my screen and introducing the speakers. So I hope everybody can see this. I'd like to welcome and thank our speakers this afternoon, Professor Oliver Craig of the BioArCh unit in the University of York, Dr. Laurentiu Angheluta of the National Institute for Research and Development in Optoelectronics from Romania, Dr. Canan Çakırlar of the University of Groningen in the Netherlands. I will be presenting her paper on her behalf, and Dr. Giovanni Leucci of the Institute of Heritage Science, CNR in Italy, who has voiced his presentation as he's unable to be here today. So I'd like to start by introducing you to the idea of IPERION and what it's all about. IPERION is a partnership. It brings together heritage science institutions from across the world, mainly from Europe, but also from beyond. This forms an integrated heritage science structure, which allows us to cooperate with each other and with archaeologists and heritage scientists from around the world. But what does that mean in practice? Well, one of the main things that IPERION HS aims to do is to provide transnational access to heritage science infrastructure. So this means you can have access to 52 providers of heritage science, that is the equipment, the techniques, the tools, and the expert in the field of archaeological science. This means you can have funded access to those institutions and the experts, which are paid for as part of the project. And you do this by putting a proposal in to the IPERION HS project, of which we'll provide more details later, but there are lots of details on the website, so please do visit that. So what can you actually do? Well, you can answer those burning research questions, and I've just got a couple of examples here of the kind of things that we're looking at. So, for example, when was your site occupied? Where did this person come from? If you've got a cemetery site, you might want to look at isotopes, and you might want to look at ancient DNA to find out their gender or their genetic makeup. So this is the kind of thing that is possible using the IPERION HS facilities. You might be interested in an object, what it was made from, how it was used, how it was made, and what other sites are present within your landscape. I do apologize for the spelling mistakes there. And also, how can you preserve objects for future generations to enjoy? So all these kind of questions that you might be saying, well, if only I could work with this person and had access to this particular type of scientific kit, I will be able to find out more about these objects, about my site, about my material. At the same time, as part of your visit, you are able to build up your competence in archaeological science because you will be working with experts and learning about new techniques and facilities and how to use them and how to interpret the results that are produced in that way. And as well, of course, on top of that, there's the possibility to co-produce research papers a result of your visit and a result of the results that you obtain from the research carried out as part of these access visits. So a little bit more about how IPERION works. So basically the specialist time at the institution is covered by and funded by the project. And a proposal can involve visits or collaboration with more than one institution in more than one country. It can also involve more than one individual. So if there's a group of you who wanted to visit a facility, so say the archaeological director and the person working on the material, that is possible. The expert's time working with you is also covered, and you may be working with more than one expert, but that one day, if you spent one day working with 2 people, that would still count as one day's access. And you, as a user, your travel and subsistence are covered. And most visits, this is not for large projects, but it's looking for a few days to several weeks. And also, in this time of COVID, virtual visits are possible where you can actually collaborate virtually, potentially sending samples to be analysed and then discussing the results online. And as I said, more is available from the IPERION HS website. So to kind of understand the way IPERION is put together, we have 3 different platforms, and these platforms are known as ArchLAB, FixLab, and MoLab. And normally you would apply to 1 or 2 of these platforms. Now, ArchLAB, the best way of thinking about this is it's basically facilities that have collections and data available. So if you're thinking about comparing your material with other material, or you want to compare your results with other results, data that other institutions might hold, then ArchLAB is kind of where you would be looking. So it's museums, comparative collections, etc. When thinking about FixLab is about scientific kit. So you would bring the material to be analysed using that scientific kit and in collaboration with the experts who understand how to use that kit and how to interpret the results. And MoLab, as it suggests, is similar to FixLab, but in this case, the instruments come to you to look at your site, look at your objects. And what we're going to do next is introduce you to some of the examples of some of the platforms that are available through IPERION HS. And at this point, I would like to invite Oliver to give us an example of a FixLab, the BioArCh facility in York.
Okay, thank you, Jill. I'll just share my screen. So hopefully everyone can see that. So just to reiterate from what Jill was saying, I think the transnational access offered by IPERION is really important for our field. I feel in the range of applications available in bioarchaeology, which is the study of biological remains in the past, quite often the expertise has been held with relatively few labs, expert labs. And there's a growing need and realization that we need to kind of democratize the science behind this and offer this service to an increasingly broad range of researchers. For our own research at the University of York, where BioArCh is based, we also find massive value in actually working closely with collaborators who know the material, who are familiar with the archaeological sites. And quite often our best collaborations come through those kinds of interactions. So actually, it's a very much a two-way process, IPERION. It enhances our research potential, but also offering the techniques that we do to the wider community. So I'll say a little bit about what BioArCh is, and then I'll give a few examples of some of the research that we're doing. So we are an interdisciplinary facility for bioarchaeology. We're based at the University of York. Quite unusually, we're actually within an archaeology department. So as I said before, quite often the techniques of biomolecular analysis, archaeological chemistry are associated with science departments. But at York, we have this unique structure where we offer this range of techniques very much from an archaeological perspective. So what do we do? Well, as I said, we specialize in recovering information about the past from ancient biological remains, artifacts, and sediments. So that includes, of course, bones, plant remains, animals, human remains, etc. from the past. But also our research focuses on artifacts. So for example, this is a middle Jomon pot from Japan where we've been working on, beautifully decorated artifact. And you might think, well, where's the biological remain in that? Well, of course, these materials can trap the remnants of food or other organic materials they've been in contact with, and we also specialize in recovering information from those. This is parchment, which is written on vellum, which is also a biological resource if you think about it. So we're able to actually analyze, in conjunction with the people looking at what was written on the parchment, we're actually able to analyze the material itself, for example, to determine the species the vellum was coming from, or even use isotope approaches to recover environmental records from the vellum. So what are we interested in as a research group? Well, we're very much interested in the evolution of human diets, and we do a lot of research onto changes in diet through time, going all the way back from the Paleolithic to the post-medieval period. We work closely with our colleagues in the environment department, and we're interested in past human-environment interactions. So perhaps how the environment has influenced human development, human culture. And we've got a big project at the moment looking at the marine environment and the impact on coastal populations, but also, of course, how more recently coastal populations have interacted and damaged the marine environment. We're interested in the evolution of disease and disease resistance, and we can get that information by looking at DNA present in bone, or indeed we can actually start to recover molecules from this mineralized plaque on tooth, this dental calculus, which will give us information of the kind of microbes that were present in the person's mouth. We also do a lot of work on the population history of organisms by analysis of skeletal remains, and sometimes more recently skin or hair remains. We have geochronologists and paleoclimate people involved with us, so we're interested in getting environmental records out of ancient biological remains, and we very much link in with people working on material culture, also doing the mineralogy or typology or other forms of material culture analysis, as well as the biological. So by definition, we have a range of people present, where a lot of us are archaeologists by training, but we also have paleoecologists, molecular biologists, geneticists and biochemists, and also organic geochemists. So we're quite a broad range of different scientists working towards these kind of goals. So we were established in 2003 with archaeology, within archaeology, but we have very close links with the biology and chemistry departments in the University of York. And we're 6 academic staff, 15 research staff, 15 PhDs. We have 4 technicians and an administrator. So we're a fairly large group. I mean, one of the largest in Britain, and in fact, one of the largest in Europe at the moment. And we have a fairly wide range of funding portfolios, going from the UK funding mechanisms, and we do also have a lot of grants coming through the ERC and Horizon 2020 programs, and of course now the Horizon Europe program. So facilities and equipment. So within BioArCh, we actually have one of the largest European vertebral skeletal collections. So another area we're interested in is osteoarchaeology. So that's actually housed within here. We have access to light stable isotopes. So Gill mentioned this briefly, but this would be to measure, for example, carbon or nitrogen isotopes present in bone to study diet. We have a range of mass spectrometers, which allows us to investigate molecules associated with artifacts or present in sediments. We have a UK Funding Council recognized dating facility, the NIRD recognized dating facility, and this is using amino acid racemization dating, and that allows us to date biominerals such as shell, so that could be mollusk shell or egg shell, also coral. And what we're able to do there is to look at these different forms of amino acids that change from one form to other at a constant rate and allow us to construct quite robust chronologies. And the advantage of this approach is that we can actually extend back beyond the limit of radiocarbon dating. We also have a Paleogenetics Lab and facilities. So we have a separate laboratory for undertaking DNA extraction and a modern sequencing lab as well. And then also, I'll talk about it in a minute, we developed the field of paleoproteomics in York, and we particularly this approach called zooms, which we use for identification of small fragments of bone. And then also we have links to other facilities very near us, such as the York Experimental Archaeological Research Centre. So that allows us to do experiments to recreate some of the materials that we see in the archaeological record as analogues to allow us to understand processes of degradation, for example. We benefit from a centre for excellence, a national centre for excellence in mass spectrometry. So we are able to use some of the mass spectrometers in addition to our own present at this facility to get very sensitive and specific identification of molecules present in archaeological materials. And then we're linking with another group which is focused on human paleoecology, evolutionary origins, and material culture studies. So I'm just going to give you a couple of examples of the kind of things we do and a little bit of the story behind this. So this is Nathan Wales, who heads up our paleogenomics group. And most of you perhaps will know about ancient DNA and the utility of ancient DNA for looking at past human populations. And we've seen large papers come out looking at the population genetics of Neanderthals. We've looked at the arrival of the Neolithic into Europe and the endemic diffusion associated with genetic change. But I think what Nathan's expertise is here is actually looking at DNA in quite novel sources. So he works a lot with botanical remains to pull out DNA from these botanical remains, seeds, grape seeds, for example, or cereals, to shed light on the genetics of domestication of those different crops. He also works, for example, on sediment samples, and we have a very neat way now of being able to take archaeological sediments and undertaking extractions of DNA from that and providing information on the diversity of plant and animal species that might have been present. Similarly, this is work on parchments. So we're by using an eraser, we're able to remove parts of the parchment and then undertake DNA analysis on that material to work out the genetic structure of the animals. In this case, it's vellum from calves that was able to contribute to that manuscript. So really unique and novel sources of DNA. I kind of run the archaeological chemistry lab as well as directing the unit. And then we're really interested in here is revealing the use of everyday objects. We do get contacts sometimes from museums, and we work very closely with the British Museum in London, and we work on some of their collections. But generally, we work on artifacts that perhaps don't seem so prepossessing for this kind of analysis. So standard archaeological pottery, coarse pots that don't seem to have much value to anybody. The stuff that's found on archaeological sites routinely can actually yield really important information regarding their use and the types of animals and plants that were utilized in these vessels. People quite often come to us with later pieces. So this would be glazed ceramics, for example. And we've also shown more recently that actually even glazed ceramics can absorb some of the residues from the foods that they were in contact with or the liquids they were in contact with. We're working a little bit on grinding stones to see whether we can recover types of proteins or lipids from these sources. And we also work on these carbonized deposits that are quite often found adhered to archaeological pottery. Here's an example, and this is the kind of routine thing we receive. This was a group from Switzerland who contacted us about this interesting barrel dating to the Bronze Age and found in the Swiss Alps. And as you can see, it had this very kind of dark residue on the inside. We said we were interested in analyzing this material as a single sample, a few weeks work, and we were able to reveal the presence of cereals within this black amorphous residue. And that's really interesting because it gives us a way to actually investigate cereal production in the past. And we now are starting to apply that much more widely to routine pots. But the molecules we're identifying, the ones we knew were surviving, originally came just through this small contract work with a group who wanted to understand what the residue was. So quite often, these smaller projects can lead on to much larger ones, and we're really interested in doing that, as well as producing a publication. And then finally, biomolecular analysis of skeletal remains. Yeah, perhaps this is what we're contacted most about. So these are the skeletal remains from the site of Herculaneum. So this is one of the boat sheds that was found near the shoreline. And you can see this is quite an unusual deposit because perhaps there are 80, 90 individuals present within this single building. And you can see there's some quite unusual things happening here. And this was colleagues at the Pigorini Museum in Rome who contacted us and said, would we be interested in doing some dietary reconstruction of these individuals to try and look at their lifestyle. And we actually have done many papers on this now, this unique collection. And we found actually that the protein in the bone, which allows us to reconstruct diet, was very well preserved, which is quite unusual because obviously the site was subjected to high heat with the AD 79 eruption of Mount Vesuvius. But not only that, we were able then to actually look at very specific differences in the diets between males and females who were in this very unusual deposit. And why that's significant is because, unlike a cemetery where we're looking at perhaps individuals who have reached their end of their lives and were buried and extends through a long period of time, this provides a snapshot of an archaeological population that we rarely get to experience in this field. So very exciting results there. Quite often we're very interested in this material and, as I said, dental calculus. And one of the things we've been routinely identifying in are the proteins entrapped within this material, as well as providing us information about the health, the systemic health of the individual. We can also recover food-related molecules, and quite often we're finding molecules associated with dairying. So telling us that back in the past, people were directly consuming milk. Now, you might think that that's perhaps not so unusual. We know dairying has existed for a long time, but the direct consumption is actually interesting, considering we also do genetic analysis of these individuals to show that they were lactose intolerant. So there's something very interesting happening between the actual consumption practice and the dietary adaptation. And then finally, perhaps what we're most known for, and there are a few labs doing this now, but we do offer this through IPERION, is our Zoom service now. And that is really taking very, very small pieces of bone, so much smaller than this, in fact, very perhaps a couple of millimeters in length, couple of millimeters in width, and able to actually release the collagen molecules from those small pieces of bone, and then analyze those using mass spectrometry to look at the fragments of the protein and assign those to a specific species. So we can now, for example, take an archaeological assemblage of very fragmented material, which may be not very useful for conventional osteoarchaeology, but we can actually get this taxonomic information from these small fragments of bone, and really helping us to understand the practices of taphonomy a lot better, but also to find those species that perhaps aren't so represented in the larger bone assemblage. So we have this as a service. It's called collagen peptide mass fingerprinting, or ZooMS, and it's routinely taken within our group. So I hope that's illustrated some of the things we do. I'm going to stop there, but of course we are willing to receive any information that you might have, any clarifications or any questions is absolutely fine. And I've put our website at the start, on the start slide, or you feel free to contact me. Thank you.
Thank you very much, Oliver. That was really interesting. I realise that we are quite short on time, so I'd like to proceed straight away to ask Laurentiu to share his presentation.
Okay, can you see me? Can you hear me? Can you hear me?
Yes, that is fine.
Oh, okay, sorry. Hello, everyone. Greetings on behalf of my team from Romania. I am Laurentiu Angheluta, researcher at the National Institute for Research and Development in Optoelectronics, INOE 2000. Before I begin, I would like to thank the hosts for the efforts into organizing this webinar. Today, I'm going to talk about a few applications regarding the versatility of one of our core tools for investigation, namely our mobile laboratory. Okay, but first, let me briefly introduce my department, entitled Optoelectronic Methods and Techniques for Restoration and Conservation. It was founded 25 years ago in 1996 as part of the Institute in INOE 2000. And our main purpose is to develop and improve the modern and scientific approaches in cultural and natural heritage protection and valorization methods. Most of our methods and technologies are based on several principles that are focused on the minimization of the physical contact with the studied surface. Our lab is certified by the Romanian Ministry of Culture, as well as our team members, the researchers within the department. Okay, one of our main tools in fulfilling our mission is our mobile laboratory, entitled Art for Art. It was designed and built in 2008. Its purpose is to bring the laboratory instruments and human expertise to the movable or immovable cultural or natural heritage assets. The case history covered with our instruments and expertise include archaeology, paleontology, speleology, and so on. Our system is modular, meaning it can be configured with specific equipment combinations designed for different case histories and application, like investigation, diagnose, monitoring, or even intervention. In the diagram below, I'm not sure if my mouse is still visible with this presentation. We can see the main methods we are employing during different situations and a few of the possible deliverables. Specifically in archaeology collaborations, we use 3D digitizations, aerial survey, geophysical survey, but they can also be complemented with physicochemical analysis or other methods depending on the situation. So far, our mobile laboratory has been all over Romania. On the top right image, I have marked a few of the cities and the sites we've already been to and performed different works. But also abroad in countries like Slovenia, Bulgaria, Spain, or Austria, with either in workshops or working on sites. To finally tackle the main topic of this webinar, I would like to briefly talk about 3 different case studies that are anchored within the archaeology field. The Roman city of Vulpea Traiana Sarmisegetuza, an unearthed 16th century Orthodox church ruins found under a great cathedral in Râmnicu Vâlcea, and a set of rupestral monuments from Bouzou Mountains. They are all located in Romania, and on the top right map, they are marked with red dots. With the yellow dot is our headquarters near Bucharest. So let's begin. I'll start with the Vulpea Traiana Sarmisegetuza site because it's located in my hometown, Hunedoara, but it is also one of the largest and most important Roman cities in the Roman conquered Dacia. I won't insist on the historical details of the site, of any of the sites I'm going to talk about. So I'm just going to briefly present what we did in each place. So in 2015, we started the collaboration project with the National Museum of Romanian History, represented by historian Ovidiu Țentea, and the local museum in Deva that managed the archaeological site, the Roman and Dacian Civilization Museum of Deva, managed by Mrs. Liliana Țolaș. Among the activities with a larger scope in this project, it spanned over 4 years. The Art4Art mobile laboratory was mainly involved in the 3D digitization, both terrestrial and aerial, of several architectural elements, as well as the documentation of the digging process of a newly found temple. Another purpose was to survey with the ground penetrating radar certain areas before they could be dug up. So in the bottom left images, we can see my colleague Alexandru surveying with the GPR, an area that was later dug up based on their finds over there. And on the right side, we have an example of the 3D model and the DM of the newly found temple, as I said. Next is an interesting private commission we had from the Archbishopry of Râmnicu Vâlcea. They wanted us to help them document and investigate the ruins of a 16th century church ruins found under the floor of their main cathedral in the city, in Râmnicu Vâlcea. What we did was to 3D digitize the whole digging site to survey the ground with the ground penetrating radar, the dig site, but also the surroundings of the church and the cathedral, and to investigate the very well preserved 3 layers of murals on 4 pillars in the old church. The walking level of the old church was somehow 1.5 meters above the current walking level. On the middle left image, you can see again Alexandru standing. That was actually the old walking level of the old church. So there we used the hyperspectral imaging and the photogrammetry to document these murals. The results are still under an ongoing study for a future scientific paper that will also include a proposition for the virtual reconstruction of the old church based on documented ruins, old documents, and similar monuments from the same region and and period. In the middle, you can see a 3D reconstruction of the whole dig site inside the church, and on the middle right image with translucid material is the beginning of the proposition for the virtual reconstruction of the old church based on the ruins we have documented. And on the right side, you can see photogrammetry results for one of the murals on the 4 pillars. And the final one, we arrive in Bouzou Mountains, a location similar with a fairy tale place. Here we find an impressive cultural landscape spread over a large area containing no less than 30 rupestral monuments dating from the late Hallstatt until the late medieval period. Our mobile laboratory was involved here in a demonstrative project for the characterization of the cultural landscape. The project was focused only on 5 monuments from the all 30. These are the 5 monuments. I won't read them. This was a multidisciplinary collaboration with a speleology and biology team from the Romanian Academy, namely the Emil Racoviță Speleology Institute. And we were, especially the mobile laboratory was dedicated for aerial survey of the areas surrounding those 5 monuments, ground penetrating radar survey for detecting any possible hidden caves or other galleries around or inside the hermitages, and of course 3D digitization of all these monuments. What we also did there, we set up an intelligent microclimate sensor network that monitored all the 5 monuments, both interior and exterior, for a whole year, for a full year. The other team from the Romanian Academy, they were focused on the natural side of the characterization of the landscape, the cultural landscape, especially on the habitat, petrographic studies, and the taxonomic diversity in the area surrounding the monuments. All these data were grouped and disseminated in an online platform. All the results, you have the link displayed at the bottom of the page. Most of our collaborations end up with a detailed report or some published papers regarding the results depending on the needs of the beneficiaries. But often enough, we also have informative meetings with them, explaining them what can we offer and how what we offer can help them. But honestly, few beneficiaries are very well familiarized with all the modern technologies, and especially with the deliverables and how can they profit from this. So depending on the contracts, we also develop complex deliverables like databases, online results repositories, as we saw earlier. Or on a different approach, we contribute and encourage the use of our results for further developments for modern exhibitions in museums or art galleries. For example, in the top left, we have a schematic from the project I presented earlier, the Artemis project. Is the schematic of the online platform that we've built. And below, there was a very interesting collaboration with a group of artists that used our 3D laser scanning of a 4th century painted tomb in Constanța, a very famous one here in Romania. And they 3D printed in 1:1 scale the whole tomb, and hand painted it with the textures provided from our 3D digitization. And the results, I'm not sure from the photo if it's very relevant, but the results were fantastic. Now the reproduction is exhibited in the National Museum of History and Archaeology in Constanța, and it can be visited there. And this is all I wanted to say for today. There is much to say about what went well and what went wrong in each of our experiences. It's been a lot of years we've been doing this, but for today, this is it. Thank you, and thank again the organizers for their effort. Thank you.
Thank you so much. That was absolutely fascinating, Laurentiu. Okay, so I am now going to present Canan's presentation. So I'm just going to share my screen so that we can talk about the facilities at the University of Groningen. So let me just do this. So I'm hoping everybody can see my screen. Unfortunately, it's gone to the beginning, so I'm just going to very quickly zoom through to the right point. So the ArchLAB in the University of Groningen includes both laboratories for conservation and materials study and zooarchaeology laboratories. They are based in the Netherlands, right on the coast down here, in a very beautiful city, which is very lovely to visit if you get a chance. Dr. Bert Nieuwbert and Drs. Gert Ormiston run the conservation laboratory under the acronym LCM. The conservation laboratory is aimed at the conservation and examination of objects and materials from archaeological sites and also from some buildings. It is the only laboratory for archaeological conservation restoration in the Netherlands housed within a university. And they have extensive experience with the conservation of metal, ceramics, organics, and stone finds, and several projects that I'm going to talk to about, and you can find out more about the projects on their website. So some of the examples of the work done here, this is to remind me that they have X-ray facilities which allow the investigation, the detailed investigation of metal objects and their associated organics, looking both at structure and also repairs and the artistry which was employed in the construction of these wonderful objects. They've also done work particularly with early architectural ceramics. This is the earliest dated example of a fabric of terracotta used as in architectural fabric, roughly 600 BC. This is from Rome. They've also been involved in the conservation of a large assemblage of Neolithic ceramic vessels excavated in the Netherlands. And this is very complicated in terms of looking at post-excavation deterioration and degradation, and how you can deal with that in terms of preserving the objects for future generations to look at, but also making sure that these objects are analysed, can be analysed in the future for studies such as lipid analysis to look at the contents of the vessels, how they were used, etc. The zooarchaeology facilities are very extensive, in some ways comparative to the facilities available at York and elsewhere. They have over 5,000 specimens of archaeological materials, so different animals, including domestic animals. And here is a case where there is a user-friendly workspace where you can work alongside the experts. And if you are interested in particular species, then you can look at this comparative material as held by this institute and also other institutes within the IPERION HS partnership. They also have an isotope preparation lab, so you can also look at isotopes, which allows you to look at movement of animals. And they have some very sophisticated imaging techniques as well. So they undertake teaching, imaging, analysis, and the lab is run by Dr. Canan Çakırlar with Dr. Shamama Vimish is assisting her and working with her on these collections. And you'll see, as well as holding vertebrate remains, they also hold mollusk remains, etc. They have a number of projects that are involved with, including a detailed project on the camels and camelids. The Sea Changes project is a collaboration between marine archaeologists and marine biologists, and it is looking at human interactions with marine life. So the history of fishing, everything from sperm whales to herring. And again, they're also working on a large project looking at the emergence of domestic animals in the Netherlands. So it is possible to consider projects that would look at and compare results from different areas within Europe as domestic animals are move into, so we're looking at the transition into the Neolithic from the Mesolithic and how that occurs and how animal and human relations interchange at that time. Oh, so this is for later, so I do apologize. I'm going to go back if I can. So this is really just a very short introduction to the ArchLAB in Groningen and the facilities available. So thank you. So our final presentation is from Giovanni Leucci on the Geophysics laboratories in Lecce in Italy. And I am going to play his presentation. So I hope this works. And please do interrupt me if it doesn't work properly. And I do hope you will all be able to hear.
Good morning, I introduce myself. I'm a geophysicist research director at the Institute of Heritage Science. I will present you the Geophysics Laboratory for Archaeology and Cultural Heritage. The staff of the laboratory is composed by 4 units: Lara Giorgi, Giovanni Leucci, Ivan Ferrari, and Francesco Giuri. The laboratory is equipped with many instruments, each of which is linked to the for example, the sonic, ultrasonic, and the seismic instrumentation that are used to measure the velocity of propagation of the seismic wave, that are to a characterization of the physical-mechanical properties of the investigated materials. We have also the geo-resistivity meter that are used to measure the resistivity, the electrical resistivity, that are to characterize the investigated material by the study of the electrical resistivity. We have also the magnetometric instrumentation that can use it also in gradiometric configuration. The activity of the laboratory are several. You can see some activity that are the integrated geophysical surveys for geological, geotechnical, hydrogeological, environmental studies, integration of physical and geophysical methods for non-destructive diagnostic of cultural heritage, geophysical methods applied to engineering, forensic science, and archaeology, and other applications that you can see on this slide. The scientific production of the laboratory are several. We have about 200 scientific publications, 400 communication at international congress, 7 books, 30 book chapters, and 3 Italian patents related to geophysical instruments. We have also several awards. You can see the awards of the last 4 years. And in this slide you can see my project related to advanced environmental monitoring system, non-invasive investigation for enhancing the knowledge and the valorization of the cultural heritage, the innovations for data processing in the cultural heritage, and we have also more collaboration with international university, such as the Australian National University, University of North Alabama, University of South Florida, and also the Heritage Malta. We collaborate also in the Pompeii Preservation Project, in Sagarassos Archaeological Project, and several other projects. And now I show you some examples of application of the geophysical method. The first case studies is at the Concattedrale di St. John in Valletta, in Malta, and aim of the investigation was to highlight buried structures, and we used the GPR method, the ground penetrating radar method, that allow use to identify the archaeological structures. This is the team that worked in Malta, and this slide you can see the results represented by the depth slice that allow use to identify tombs and wall. You can see this as a virtual excavation that allow use to see at several depths in the subsoil. But the 3D surface representation allow use to well see the buried structures such as tombs and wall. The second case study is the archaeological site of Sagarassos in Turkey, and the site is the Roman thermal bath. And the aim of investigation was to highlight the cases of deterioration of the structure of the Roman bath. And here we use the ART method, electrical resistivity tomography, that allow use to identify effectively the deterioration causes. This is the investigated area on the Roman bath, and this is a 2D section that show the resistivity at several depths. This is the Roman bath, and this is the roof of the Roman bath that show some zone of water filled and several fracture present on the roof. This is a pseudo 3D representation by depth slice of resistivity, and in this case we can see the water flow, this blue area, at several depths, and then we can follow the water distribution of the roof of the bath at several depths, and this is a zone of accumulation of the water, and this is a 3D representation by resistivity subsurface that show the water flow on the roof of the bath, and this water is the cases of the deterioration of the roof of the bath. Other case studies is the application of geophysical method at the archaeological site of Pompeii in collaboration with the Carabinieri of the Protects Cultural Heritage. And the aim of investigation was to identify clandestine excavation, and in this case we use ART and GPR methods, and the results are used to identify the clandestine tunnel and archaeological structure. This is the investigated area, Via Civita Giuliana, and it is a very famous area. And this area is about 2,000 kilometers from the archaeological sites of Pompeii. These are the photo of the acquisition data, and this is the investigated surveyed area. This is the main results that of GPR survey that are used to see some structure such as wall or room, and this dashed yellow line represent the roof of the tunnel. The tunnel is well visible in this representation, and you can see also in this representation as depth slice the tunnel. And in the 3D isosurface visualization you can see the two rooms and the tunnel near the two. This is the 3D earth depth slice that shows effectively the path of the tunnel and the other archaeological structures. These are the photos of the excavation results that effectively show the tunnel and some archaeological structure. See on the surface the amphitheater, but geophysical investigation to identify all the structure of the amphitheater. You can well see in the 3D isosurface representation, 3D topography representation. Thank you very much for your attention. If you have any question, you can send me an email. Thank you very much.
So just to wrap up and to just reiterate how to put together an application for transnational access. So this is the third call for proposals, which is now open. The closing date is the 30th of April 2022. Please do watch the videos on the website that tell you how to put a presentation together, and contact the help desk by email. The information is there. They are always happy to help with questions, and they can put you in contact with experts who can help to refine your proposal, but also work out what instruments you might want to have access to, or whether there are particular archives available that you might want to look at. There are also help desks for each of the platforms. You can put an application in for transnational access at any time, but there are 2 evaluations proposal of the year linked to the 2 calls per year. I should also state that the reason why it is called transnational access is because you cannot access IPERION infrastructure in the country in which you are based. So me being based in the UK, I cannot access infrastructure within the UK, but I could access IPERION infrastructure from any of the other partners in Europe and beyond. So I think that just gives you a wrap up, and we would welcome any questions from our participants. So I will leave the floor open to our participants.
Thank you very much, Jill. It was a really, really interesting session, an excellent webinar, and I learned an awful lot, I must say. So this completes our 2021 webinar series within the Heritage Science Academy. And I'd really like to thank our colleagues in the central office for having organized the series and enabled the series. And as you probably know, all of our webinars are available as video recordings on the E-RIHS and IPERION HS YouTube channel. What remains for me to do is to make a short announcement of our very, very exciting 2022 webinar series. We're not in a position to announce the names and titles yet, but to whet your appetite, I can announce that there will be webinars on science topics such as laser techniques for heritage material characterization that are available within IPERION, as well as the recent developments in this field. There will be a webinar on hyperspectral and multispectral imaging. There will be a webinar on olfactory heritage even, and how research infrastructures could help there. And there will be policy and other webinars as well. For example, we will be listening to how S3 infrastructures are evaluated and monitored. So hopefully something in our program for everyone. So what remains to be done is to thank Jill and our speakers of today, and to all our participants for having attended this webinar. Thank you very much indeed.